Okay, we're at 11 a.m. Pacific time and 2 p.m. Eastern time. Thank you everyone for joining us from all across the globe for our webinar today. So this webinar is brought to you by the team of Plant Method Exchange Platform, our PMAX. So our team was established based on the North American Epilepsy Steering Committee, NASC. And then we expand our team through the year. The primary goal for our platform is that we want to have a free open access platform to organize and curate published and unpublished shared protocols so that people can carry out their experiment with ease. And in the meantime, we also want to have interactive platform for people to discuss and connect with experts who have expertise in certain areas. So that's one of the reasons we have this interactive workshop series. And here is our team members. So currently on our leadership team, we have Mingyuan Zhu, now postdoc in the Benfei Lab in Duke, and soon to be assistant professor at Texas A&M. And I'm Xiaohui Li, I'm a postdoc at Caltech, and Jianqiang Shen, assistant project scientist at UC Berkeley, and Mingjie He, postdoc at Duke, and Vincent Cervantes Buenu, postdoc at UC Riverside, and Catherine Freed, postdoc at Wisconsin Madison. So for our workshop series, our scope is to bring together experienced researchers to share practical insights and lessons learned from applying scientific research techniques in plant biology and aim to foster deeper connections across the plant science community. We have a series of workshops lined up. Today is our first workshop of the series. Today we're going to talk about morphogenetic regulator-assisted plant transformation. And we have 3 panelists today that I'm going to introduce shortly. And our next topic is quantifying plant morphogenesis in 4D using morphographics. We're analysis of confocal images. That is, our target time is September 2025. So look for our notifications in the future. Remember to register if you are interested in this webinar. And the third one lined up is what happens after translation, exploring post-translational modification. We're gonna have that in early December 2025, and we're gonna have a few more next year. If you have some topic in mind, you're also welcome to suggest to us. So in today's workshop, we have 3 panelists. Dr. Jianqiang Shen. Dr. Shen is an assistant project scientist at the Department of Plant and Microbial Biology at UC Berkeley, and he also holds a joint appointment at USDA. Previously, he obtained his PhD from Huazhong Agricultural University in China and worked as a postdoc at University of Pennsylvania and UC Berkeley. And we have Dr. Juan Manuel Debernardi. Dr. Debernardi is currently the manager of the plant transformation facility at the University of California, Davis. Previously, he got his PhD from National University of Rosario in Argentina, and he worked as a postdoc at UC Davis. And our next panelist is Dr. Greg Goralogia. He is currently a postdoc research associate at the Department of Forest and Ecosystems and Society at Oregon State University. Previously, he got his PhD from University of Washington in Seattle. All 3 panelists have extensive experience in developing transformation methods for multiple plant species. This event, we're very glad to have all 3 of them to share with us their expertise. And after the webinar, we're gonna have a live Q&A. And after live Q&A, if you still have something you want to discuss with the panelists, all 3 of them, they are very happy to connect through email. So feel free to send them email them further questions. This event is brought to you by the Plant Method Exchange, or PMAX, leadership team. In the meantime, this event is also co-organized with Plant Postdocs, a grassroots organization that formed and run by postdocs in the plant biology research field. And this event is powered by NASC, the North American Epilepsy Steering Committee. And thanks again for all of you to join, for all the panelists and attendees to join. So we're gonna start the first part of this webinar. So each of the panelists, they will give a presentation. And just as a reminder for the Q&A, so if you have questions, please put in the Q&A box. Don't put in the chat, as it's difficult to track the chat. Please put in the Q&A. And with that, the stage is yours, Jianqiang.
Sorry, I was muted. Can you hear me? Cool. So, hi everyone. Good morning and good afternoon. Continue to share. Is that good? Cool. Thank you, Greg. Okay, so let's start. So today, thank you for the introduction, and I'm so happy to be here and sharing our efforts on sorghum transformation to development and also editing to optimization. My topic is that it will be the high efficiency transformation tool used to expand the editing scope in sorghum. So let's start with sorghum first. Sorghum has been identified by the DOE as a flagship bioenergy crop, and also it is notable for its high productivity on marginal land, even under the climate-driven conditions such as low water and nitrogen availability. Sorghum used as a food stock, feedstock, as well as a biofuel. And recently, we consider sorghum as a carbon sequestrator. So at UC Berkeley, Peggy Lemur's lab, along with Chris Newgie's lab and David Savage's lab, we are working on several projects to improve the sorghum photosynthesis, as well as to increase the root biomass of sorghum. So let's move next slide. But sorghum is also considered as a recalcitrant crop, which means it's hard to get transformed. Let's walk through the whole picture of the cereal genetic engineering. So I think it's about 35 years ago, Peggy and her colleagues developed the first maize transformation using bombardment. And then 2 years later, Mary Dale Chilton, who developed the agrobacteria-mediated transformation, it's also in last century. And then the people who is working on cereal transformation, they found that the embryo might be a great explant for transformation. But since then, there's no big progress on the transformation area until I think it's about 2000. A group of basic researchers, they found that morphogenic gene regulates plant cell totipotency, including baby-born Wurshow. It took about 15 years to get it to work in maize transformation by Bill Goldenkamp at Corteva. So since then, we started follow up and apply this technology in sorghum transformation, and we get it to work and publish it in 2021. It took a long time. So I believe today, Juan and Greg, they will share more interesting story about the morphogenic gene assisted transformation. So I'm looking forward to hear that as well. Okay, so let's work through the conventional sorghum transformation. So we use immature embryo, as we talked about, and it's isolated from the seeds and infected by the agrobacteria. It took 1 week co-cultivation, 1 week resting, and 4 to 5 months for regeneration. Yes, that's right, 4 to 5 months. And then we developed this construct using baby-born Wurshow, but the other one thing is integrated baby-born Wurshow might cause developmental deficiency, so we have to excise it after transformation. So applying the baby-born Wurshow, we still the same, 1 week co-cultivation and 1 week resting, but it would short half of time. The regeneration it reduced to like 2 to 2.5 months, so which is great. And it not only reduced the regeneration time, but also it would improve the transformation frequency from less than 10% to more than 50%. And another great thing, it also would reduce the genotype dependency, at least in sorghum and maize, which is a great job. But in sorghum, there's one limit, which is the knockout efficiency. So previously in our paper, we reported it's about 17% editing efficiency, but we would like to improve that at least to some level, which we can use them. Okay, so we developed this like shadow and binary vector. So in shadow vector, it includes the the Cas9 gene, as well as the guide RNA expression cassettes, including the polycistronic using tRNA as a spacer here, and also monocistronic, which means one guide RNA driven by one promoter. So this construct, as long as we insert our own or customize the guide RNA, we will use them in for pro-positive transfection after validation, and we will move the whole expression cassettes to the binary vector. So here's all the binary vector we developed, and the first one that was the basic backbone, just the LR recombination site, as well as the hydromycin selective marker. And also we use this screen vector, which is used for screening the morphogenic genes in sorghum and maize. And also ultimately we developed an editing construct, and previous reports said the virtual integration might increase the transformation frequency as well. So we use that, we took advantage of that. So we use the virtual expression cassette, and then we add a ZS screen expression cassette as a visual marker, which is used for tracking tDNA as well. So then we developed two constructs, and as I shared with you, previous study showed that the polycistronic of the guide RNA and the monocistronic might lead to the different editing efficiency. So we tried both. So we used two guide RNA targeting the PDS gene on the exon 3 and exon 4. We transformed into sorghum. So in this case, by integrating the virtual, the regeneration time can even reduce to 1 to 2 months in total. So we use 222 immature embryo for polycistronic, and monocistronic is about 205 immature embryo. We got 69 regenerants and 50 respectively. And by genotyping them, we got about 22 edits out of 69 genotyped transgenic plants, and the editing efficiency for polycistronic that is about 32%. For monocistronic, it's even higher. It's almost to reach up to 96%. So here's take a close look at the genotype, and the majority of them, they are one base pair or two base pairs SNPs. And here is, and we also define the knockout score and as a proportion of the indel that indicates a frame shift or are more than 21 base pair in length, deletion or insertion. So here is the knockout distribution of the guide RNA, which is pretty good. And after that, we are thinking of using the different different nucleases to at least to expand the editing scope. So we are thinking of different options like Cas12, Cas9 variants, and etc. So we found that Sprite might be a better option for us. So we mutagenized the de novo from the gene we have, a maize Cas9 with one intron. So also use the same guide RNA targeting the same genes. So we got about 49 regenerants out of 109 immature embryo, and 46, and 49 out of 117 immature embryo for monocistronic. And interestingly, we saw the excellent phenotype of those edits, which is a complete albino or chimeric albino phenotype. So here is the editing efficiency, and for the polycistronic, it's about 25%. But for the monocistronic, it's about more than 80%, which is pretty good. And here is one more information, and I would like to point out here for monocistronic, at least on the guide RNA 2, it might cause a larger deletion. Larger means more than 5 or 6 base pair. So which is kind of new to this Sprite editing events. So for Sprite, there's one benefit which can use as a editor for AT-rich region, and we know of that AT-rich might be in the promoter or the untranslated region. So we designed 6 guide RNA targeting the PDS promoter as well as on exons, and all of them they end up with an AT, which is pretty good. And we use 6 different promoters for driving them. And after transformation, we got large deletion, particularly on the promoter region. So we thought that might be a great idea, a great candidate for promoter editing. So in total editing efficiency, that is 58.8%. Okay, so here is a summary. So we improved the editing efficiency from 17% to 96%, at least in monocistronic. We also developed a sprite editing system. And the last slide, that is a take-home message. So we utilize the shuttle and binary vector for genetic engineering, and also developed the internalized Cas9 for high efficiency genome editing, and implemented the internalized sprite to facilitate the editing of AT-rich region. That's all for today. So I would like to thank you, Peggy, and being my advisor, and also the co-workers, Kiflin and Sultana. And also, I would like to thank all the talented undergraduates. I'm so happy working with them. And also all the lab members, Tamara, Emma, Dasha, and Sunny from Lamo Lab, and Chris Newgi's lab, and also Savage Lab, Laundry Lab, they are all my collaborators. And also, I was partially supported by the USDA, Roger, and he is a great collaborator, and also Yonggu, and who is my USDA advisor. And also, I have collaboration with Schroeder's lab, and also thank you for the Cartiva for their discussion, helpful discussion, and the UC Berkeley facilities. And also, this is the planning agency, and which is listed here. And that's all. Thank you.
Thank you, Jianqiang. Very nice talk. I see we're starting to get questions. Just another reminder, please put your question in the Q&A box. We'll get to all the questions at the end of all 3 panelists' presentation. Now we welcome Dr. Juan Debernardi to start his presentation. The stage is yours, Juan.
Okay. Thank you first for the invitation. So I did my PhD in Arabidopsis, so I'm happy to talk to the Arabidopsis community. And one of my motivations during my PhD was to translate what we know into crops. That's why I did my postdoc in wheat. So I will be happy to help and support anybody that want to do some translational research into crops. So I will talk and give some information about a technology that we developed. I think we have already a good introduction, but we developed this technology that is similar to baby bushel that we call CRF shift. And again, I will try to go quick in this part because we don't have too much time, but basically, when you want to do transformation, or when you want to do genetic engineering in crops, transformation and regeneration are an important bottleneck, and there are 3 things that affect that. That one is the efficiency. Most crops and protocols have low efficiency. Only a few genotypes can be transformed, and also labor and space is a problem. So I will go fast here because I think the previous introduction were good. So we have, I'm very lucky now in the plant transformation facility because we have a lot of expertise in cell biology and tissue culture, thanks to all the people that have been working on that field for over 50 years or more. So we know a lot of conditions, hormones, and now we are starting to know about the molecular pathway and genetic pathway that control regeneration. So we can use all these tools to improve and help during the regeneration. So I will focus now in the CRF shift technology. This is interesting because it's an example of translational science from Arabidopsis. So I did my PhD studying these genes in Arabidopsis in the laboratory of Javier Palatnik. So we were studying the regulation of GRF transcription factor by micronettrin 86, and this transcription factor form complex with a coactive vector called CRF interacting factor shift. And this complex we found in Arabidopsis that controls cell proliferation and meristem homeostasis. With Javier, we had the idea of putting the two genes together in a single transcript in order to have the pre-assembled complex. And we found that when we did that in Arabidopsis, we have a boost in the activity of CRF with more cell proliferation, and that also affects the meristem of the plant. So when I came to UC Davis to do my postdoc in Jorge Dukowski lab, wheat transformation was a big problem. So the efficiency was low, and it was almost impossible to do efficient CRISPR on wheat. So at that time, I was working with David Tricoli, the previous manager of the transformation facility, and we decided to incorporate the CRF shift vector into the wheat transformation protocol. Similar to sorghum, the wheat transformation protocol, the one that we use, start with immature embryos. So I would say that immature embryos is a great explant for many of the cereals, and I highly recommend using that because it helps a lot and is very efficient. So, and it was very simple because we incorporate CRF shift in a agrobacterium transformation in a binary vector. And what we observed when we transform with that vector was this. So in a regular transformation, only a few embryos can produce shoot, but when we use CRF shift, the efficiency is almost 100%. So similar to the story with baby boom bushel in sorghum, CRF shift reduce the time, so and also limit the constraints that that affect transformation frequency, like plant conditions, embryo size. So that simplifies a lot the work in our transformation facility, and also help people that decide to establish a transformation protocol in their own lab. Because if you can reduce the constraints or variance between or the effect of plant quality, stuff like that, it's easy for more people to establish this transformation protocol in their lab. I will show some example of that later. And the other thing that makes CRF shift interesting is that the plants that we got after just incorporating this casein in the binary vector were fertile. So we don't need any weird or complicated expression system to control them. So something very important for us and for the wheat community about CRF shift was that it allowed us to expand the genotype that we can transform. And here I will show examples of the work that we are doing in the transformation facility. Initially, we started working with Kronos because that was only our wheat transformation services. But now we are transforming wheat lines from many different breeding programs in the US, including UC Davis, Idaho, LC Star is from Lima Grain. So we have cultivar from Montana, from Nebraska. And we also have a collaboration with CIMMYT where they were able to transform elite cultivars. And something interesting that I want to mention here is that we are starting to transform diploid wheat. So, and this is important because as a research tool, and I have this conversation many times with Arabidopsis people, that people feel like that you cannot do basic science in wheat because of its hexaploid or tetraploid, and the polyploidy, it make it everything more complicated. So now we are transforming diploid wheat. So monococcus is a domesticated wheat that is diploid, and that could be an excellent model for research. We also, something that I want to mention here, so that in addition to the morphogenic genes like CRF shift, or in the case of baby boom bushel, in our case, I highly recommend using ternary vectors. There are great tools in AdGene that you can order them and get them because these ternary vectors enhance agrobacterial infection, and they really help to expand the genotype that you can transform. So using the system, we are also now transforming several barley genotypes. Here are examples of different cultivars from different places, different breeding programs. So now I want to mention something about genomediting. So basically, we can put together in the same tDNA CRF shift with CRISPR, which in this case is Cas9. And we have a vector that is available in Shen. So this vector contains Cas9 CRF sheaf, and you can clone guide RNAs in that position. So guide RNAs can be cloned as single or multiple guide RNAs. And here I recommend the tRNA approach. In the previous talk, we also observed that the tRNA approach was used for multiplexing. That approach, in my hands, works very well and works very well in multiple species that we transform in the transformation facility. So this vector is available in Shen. And what I mentioned here, there are many other models. Something that I want to say, I'm switching a lot of our clones to the Moclo system because I feel like there's a lot of resources, models already in Shen. Cloning is very simple. And so I'm trying to move everything into that. We have generated base editors using CRF sheaf. We have also generated and test prime editing vector in wheat, and all those work very well. So we have them as a Moclo models. There is also work from the Shonin Center that they have combined CRF sheaf with Cas12, and those vectors are also available in Shen, and they work very well. So now I'm going to show a quick example of what we can do combining CRISPR and CRF sheaf, and also expanding the genotype that we can transform. So Josh is the wheat breeder. He wanted to edit triticale, that is a hybrid between wheat and rye. Triticale yield is much higher, and the biomass is much higher than wheat, but has a lodging problem. So Josh wanted to make the plant more compact. And with the technology that we have, we were able to transform triticale to edit a particular gene. So here is the micronebite inside of, so it doesn't matter. So, but we edit a particular gene that allow us to control plant height with a very precise, so very precise height. And that was very fast, and we were able to do this in a breeding line, so directly working on Josh breeding line, and we were able to put them in the field already, and you can see that the edit material is standing while the wild type material, the original breeding material, has the lodging problem. So in addition to CRISPR, people is also interested in transforming regular vectors. When you transform a regular vector, sometimes you don't want to have CRF sheaf in the same tDNA because CRF sheaf plants are fertile but can have some effect on the analysis. So one way that we do that is by doing co-transformation. This is similar to the altruistic approach that you will see later, and also with baby boom bushel. And doing co-transformation, basically we co-transform two agros. You can get the high efficiency with regular vectors. So it's important to play a little bit with the ratio of the two agros, but that works very well for us in wheat. So in terms of other species, I don't know how I'm with the time. I'm okay, or?
It's okay.
It's okay, okay. So I will mention a little bit what have been done in other species. So this is mostly work done by other groups, not by us. So it's great for me. At least I feel very happy that I see that other groups can replicate what we have done. CRF sheaf was validated in several crops, in several monocots, including maize, sorghum, oat, rice, and also in several dicots. I saw that in the audience is Lawrence. So with Lawrence, we published together. So Lawrence led that work using CRF sheaf in maize, and also he combined and he showed that using CRF sheaf with ternary vector actually make a big effect. And then there are multiple other examples of the use of CRF sheaf in dicots. As I include here, and as I mentioned before, we have developed Moclo models for several CRF sheaf from multiple species. We have CRISPR vector and regular binary vector that are very easy to clone, that if you need something, you can contact me. I will share with you, no problem. But the idea is that you can have easy to clone vectors, and those are available. So we use most of them for our service in the transformation facility. So when people contact us, we already have vectors that we know work in strawberry, tomato, pepper, citrus, lettuce, but the crop that we transform. And these are just very quickly a few examples that we have in dicots. This is the work that we did in citrus. We actually show that CRF shift from different species can work in citrus, either citrus CRF shift or grape CRF shift. We also were testing tomato CRF shift in pepper. That is a very recalcitrant genotype, and we were able to induce a nice frequency of shoots. And we have also interesting results in tomato. There is a paper that was published, I think, this week from in tomato that shows the CRF shift also improved transformation in tomato. It's not our work; it's a work from Sebastian Soy lab in Switzerland, and more or less they were able to reproduce or to get similar results as we got. And finally, I want to show this tool that some people ask me, okay, what happens if I want to have a CRF shift only active during the tissue culture and I don't want to have a CRF shift active later, stuff like that. So there are tools to control CRF shift activity. For example, this is a dexamethasone inducible version that we validate the activity of this tool in Arabidopsis, and also we have the tool working in citrus and in another crops. So basically, we clone the glucocorticoid receptor between CRF shift, and with that, you can control just by adding DEX into the media. So we observe that DEX for some crops like tomato could be toxic, but the system works very well in other crops, and there is also a tool from the Shonines in wheat. Okay, so with that, I want to thank my team at the transformation facility. So we mostly are focused on service. So we transform a lot of crops, but we try to do, and we want to do more collaboration on research to expand genome editing tool or transformation tool in different crops. I want to especially thank David Tricoli. So David is a colleague and a friend, and I'm very lucky that I met David a few years ago, and he introduced me into this work. So, and also I want to thank other collaborators like Jorge, Javier, and breeders and PIs from UC Davis that where we interact to use this technology to improve crops and stuff like that. Okay, that's it.
Yeah, thank you, Juan, for sharing the knowledge, method, and also sharing the resources. Thank you. Next.
Let me stop share.
Yeah, next coming up is Dr. Greg Goralogia. So welcome, and the stage is yours, Greg.
Thank you for the organizers for allowing me to present today. Everybody can hear me? Good. Yes. Okay, great. So I'll talk to you about a morphogenic system that we've been using in tree species, and I'm calling from Oregon State University. So we work in forest tree species, for the most part, members of the genus Populus, a lot of hybrid poplars, and our stakeholders mostly care about hybrid eucalyptus. And these have a lot of problems for biotech, really long testing cycles. In the middle here, I think, are the first CRISPR-edited trees. And they're in year 8 in the field, and we edited floral traits, and it's the first year that we're actually getting data. But these are usually wide interspecies crosses, long breeding programs, and clones with lots of regulatory problems. So I just thought I would put this here to contrast this with row crops, because sometimes people forget. Typically in a row crop, you might use agro or some other delivery agent, regenerate, figure out what editing you might do, and then self-pollinate to get rid of the transgene, as Juan talked about in the previous talk. Then you might have edited transgene-free plants and be able to do field testing or something like that, depending on what country you live in. But in trees, most of these are clones, and they just have really challenging in vitro biology. And so both for morphogens as well as for CRISPR, this is really a problem because it's really hard to get those genes out or not get them in the first place. And as I mentioned, tree species are just hard. They're often these clones, so I don't have access to juvenile tissue like seedlings. Every genotype is very heterozygous, so it's a new adventure in vitro. And there's just lots of strong defense responses. This is eucalyptus. So the left panel is a regenerating control. You add antibiotic in and you see the cells getting killed, but you add aggro in and the cells just die. There's these really strong necrotic responses. And this is common in a lot of species. So if you think about how to tweak this, we often think about changing hormones in tissue culture, and that will get you kind of far. But I've been working a lot with morphogens like the other two speakers, which are born in a piece of DNA. And so this is kind of a modality shift about how you're exerting regeneration outcomes. And we've worked with a lot of morphogens. Woody dicots are hard, and a lot of these, particularly Wuschel, baby boom, and WOX genes, often really suppress regeneration in our system. So my focus has actually been on genes from Agrobacterium, which seem to work better in some of our woody dicot species. So this was a bit of a boondoggle, but at the start of this project, we tried to use in planta greenhouse methods, figuring that we might have kind of natural regeneration processes at different inoculation sites, a couple of different species that we tried. And really, this was helpful in us figuring out what morphogens might be doing when we injected different areas. And one of the star performers was these hormone-producing genes from Agrobacterium. And we could get reliable transgenic tissue development in a variety of different genotypes, about half of them in both Trichocarpa and eucalyptus. Sorry, I think there's a train passing by. Hope it doesn't get bad. But we could really never get transgenic shoots in the system. And so in particular, we wondered whether we could find a way to get these genes, these hormone-producing genes, and tweak them into something that would be usable in a system. And one reason I was attracted by these is that it forms this natural feedback loop. There's two auxin producing genes and one cytokinin gene, and they self-regulate to form undifferentiated cell growth. So we look back in the literature, and actually in the 90s, several groups in France had found wild Agrobacterium strains that produce shooty gall tissue. And so we wondered whether this might actually be something that was more developmentally flexible and something that we could use as a transformation system. But a lot of their funding was cut in the late 90s due to GMO fear-mongering, and really these were unwieldy at the time to try and clone. So this strain in particular actually was sent to us in 1993, and it was still in the freezer, and I was able to resurrect it, which was pretty cool. I cloned out a 10 kb region from this strain, which included 6 genes in the tDNA. And we transformed it into hybrid poplar. And you can see they formed shoots, but they're non-transgenic, sort of on the border of callus tissue. So that was pretty cool. And we were talking with people at Corteva, and they recommended this altruistic approach. Juan mentioned this. This is where you mix agro strains together. One carries your gene of interest, and the red one is your morphogen. And the idea is you get this like distribution of cells, and the red infected cells sacrifice themselves. That's the altruistic part. And you might get a plant that only contains your gene of interest. So this is like the holy grail for us in a clonal plant, because we don't get integration of a morphogen that's going to cause pleiotropic effects. So we tested this system. This is like 6 genes, as I said, a 10 kb region with a spec-resistant tDNA in the other strain. And we mix these in equal ratios and then use spec on hormone-free media. So we actually were able to see shoot regeneration. And if we look at the morphogen, it's in these callus areas that are at the upper part of the explant. And then distal to there, we get shoot regeneration, transgenic shoot regeneration that only contains our trait tDNA. So that's pretty cool. We tried different ratios. As Juan said, this is something that you can do to check. An equal mixture is what worked for us with leaf tissue in particular. And we tested this against our normal hormone-based approach, and it was a little over twice as effective, but it was also really, really fast. And so we can now get transgenic shoot production this is an amenable genotype, but we can get it within 2 months. Right now we have this strain, it's launched from the Vir plasmid in a system called Gantry. That's been a game changer for us. We also have some binaries, so we hope to add these to add genes soon. The ratio really matters. This is a critical part of this. And spectinomycin also in our dicots, because you're not killing the callus tissue as much, this is a really important aspect of it. I'll talk a little bit about the mechanism. We've been wondering how exactly these work. So originally we thought it's probably these hormone genes, and so those are highlighted in pink, but it could be some of the other genes in the tDNA. So we've done some work on this. So in order to test this, we cloned out the hormone genes from this shooty strain, as well as the reference agrobacterium tDNA, C58. So that's shown here in the middle, sort of excluding the other genes that are in this region. If we just look at transgenic callus, actually the C58 genes are better than this strain in hybrid poplar. But in terms of transgenic shoots, ones that only get the trait tDNA, actually both of them are non-functional. So you need the 6 genes and not just the hormone genes, which was pretty interesting. So that made us think, yeah, it must be these other 3 that we know almost nothing about. So to test this, we generated stop codons that are present in each locus. Those are those purple bars that you can see off to the left and right. But kept the gene orientation that's the same. That actually ended up being kind of important. And we expected that there might be some additive genetic effects where multiple genes are at play, but really just the 6B gene that's to the right of the IPT gene totally removed the ability to form these altruistic shoot production. So the 6B gene is the most critical one. This is what those look like. You can see in the 6B mutated ones that the callus actually looks remarkably different. And the rest of the various constructs were still capable of forming these GFP-only shoots, which is pretty cool. So what's unique about this? We're still really working on the mechanism. If you look, this is an alignment. 6B genes are really common among agrobacterium strains, and it actually has this unique region in the N-terminus that is this extension of basically a new protein domain. These are actually from BioVar3, which is Agrobacterium vitis. So it's a recombination event from very distant agrobacterium strains. A little bit has been worked on these 6B because it's present in naturally transformed plants that have had tDNA introduced in their evolutionary history. Right now, the sort of predominant mechanism that's been worked out is suppressing argonaute serrate, so suppressing microRNA biogenesis. But we really have no idea how this concerts kind of a non-cell autonomous regeneration effect. So we're still working out on the mechanism.
I think we lost connection with Greg. We'll wait for 1 minute. So while we wait for Greg to reconnect, okay, Greg reconnected.
They're doing electrical work in my building. I hope they didn't boot me off here. Can you see me?
We can see you and your slide now. Yes.
Great. Yeah, so I don't know when I cut out, but yeah, the basic idea is that, naturally agro strains probably are manipulating the auxin-cytokinin ratio like we do in tissue culture. So one thing we're working on is, can we sort of change that balance in addition to the 6B gene and create a suite of vectors that people can use across different species? And then in addition, we're working on both wild agrobacterium strains that are part of a collection, as well as trying to characterize some of these other genes that we basically know nothing about that might be useful for transformation. And we now have hundreds of sequence strains that we can mine for some of these. Oregon State has a lot of specialty crops, often clonal. So we started work in hops. We've also sent this strain externally to about 20 labs. We finally have a system working in a US hop cultivar Cascade. And it's a very, very tough system in the Cannabaceae, but we have ongoing collaborations there. If you want to try it, we're happy to share, and we should have a binary available on AdGene in the coming months. So in summary, these unique tDNAs from different strains can be useful in altruistic transformation in woody dicots. We still don't know how 6B is capable of this non-cell autonomous shooting effect, but we're excited to figure it out. And we hope that this might be a system in clonal crops because you don't integrate the morphogenic tDNA, and that's really critical in some of these species. A couple of points of advice from my learning. I like to piddle. I like low replication experiments that kind of look for qualitative effects, like help you read the tissue culture, see which ones are providing a beneficial effect before you commit to some huge experiment. Work on your tissue culture. It's very important when you're doing a new species. Back to basics, really trying to figure out if regen or DNA delivery is your problem. And if you can screen many genotypes. In eucalyptus, when I showed you in that first slide, that's the best one out of 100 genotypes we screened. So, you know, there's a lot of challenges in working in some species. So with that, I'd like to thank my mentor, Steve, Kathleen, and Kate, who do most of the tissue culture work in our lab. And so
Okay, we lost the connection with Greg again. When we wait for Greg to reconnect, if the audience have any questions, please start typing into the Q&A box. We also have collected questions from the attendees through the registration form. Okay, Greg has reconnected. We lost you at the acknowledgement.
thanks. I don't know what's happening here. Yeah, they're redoing our HVAC in the building, and who knows what's going on. Thank you.
Yeah. Okay, thank you. All right. We thank all of the three panelists, Jianqiang, Juan, Greg. We got a few questions from the live attendees and also from the registration form. The first question is directed to Jianqiang, and other panelists also feel free to join. So the first question is, do you see chimeric edits or transformants? And if so, how do you handle those chimeric edits and transformants?
Yes, we saw at least in T0, but for the sorghum, we have genetics. We can purify the genotype in T1 generation. This is typically it's for all cereals, and they can pass to the next generation, and then we will get the tDNA-free homozygous of edit. Yeah, that's my answer.
Okay.
Yeah, that's a very common situation that I observe in our working wheat and other crops too, service. Actually, sometimes having a chimeric is quite useful because from one plant you can have a very large population of edits in the T1 population, and so it's not always a problem to have a chimeric. Sometimes it's good.
Okay. Our next question is for Greg. So Greg, could you expand a bit on why using SPEC is important in your system?
Yeah, so spectinomycin and there are a few others that do this, primarily target plastids. And so if the tissue itself is kind of etiolated, there's not as strong of a killing effect. And for these altruistic approaches, if you need to only select for the cells that get your trait tDNA, but you need to keep the ones that get the morphogen alive for 2 or 3 weeks in culture, then you don't want to kill them too early. And antibiotics like hygromycin that are really strong in the callus phase, those are going to negatively impact the kind of morphogen only integrated cells. And so the balance of those is actually quite critical. Sulfadiazine, I think, is another one in the same class. But I think the Wisconsin Crop Innovation Center in dicots is using SPEC a lot now too. So if you're in a dicot, I would encourage you to try it out. You know, most of the old school vectors are going to be Canhig or Basta, and especially with morphogens, we found that SPEC is a better one to use in dicots.
Our next question is toward Juan. So can you elaborate on the utility and limitations of the inducible GR-DEX system for the purpose of regenerating normal plants? Is DEX toxic to some species in tissue culture?
Okay, good. Yeah, so the utility that I found for that is that because when I say that when we say that GRF shift plants have normal phenotype, normally what we are trying to say is that the plants are fertile and that you can get offspring or T1 generation without problems. But I mean, I studied GRF shift during my PhD, and those plants are bigger, they have phenotypes. So, and if you are studying something, a molecular pathway that will control development, so you don't want to have GRF shift active there. So that's in those situations, having an inducible version can be useful. And as I say, we have this version that we have tested in Arabidopsis and in different dicots. And I know the Shonines just published a different DEX inducible GRF shift. Instead of cloning the GR domain in between GRF shift, they put upstream in the end terminal of the GRF gene. And they use that because they found that in some, I think in some tetraploid wheat, GRF shift produce some sterility. So with that, they can control that. But what I say about the DEX being toxic, we found, for example, that tomato doesn't like DEX. At least you had to use very low concentration, which could be a problem when you had to induce. And I was asking tomato community because DEX is a very useful system in Arabidopsis, and I was wondering how people in tomato is doing DEX inducible approaches, but I couldn't get a good answer on that. But we do see some reduction on shoot induction when we add DEX into the plates in tomato.
Okay. The next question is also about chimeric transformance. So does keeping the callus selection media for a long time, like 2 to 3 months with subculture every 4 weeks, does that reduce the number of chimeric transformance?
I mean,
well,
yeah, go ahead. Sorry.
Go ahead. No, I'm fine. Yeah.
No, I think depend a lot on the crop that the answer, because for example, in our case in wheat, barley, or so the explant, the immature embryo make a little bit of color, but immediately produce embryos, somatic embryos. So so there is no that long phase of Kali induction, which I don't like to have the materials in Kali induction during a long time, because that you can also induce a lot of somaclonal variation. So I prefer a faster protocol that will induce embryos quickly, and those embryos can be chimeric, but most of the time what we are seeing, if you have a very efficient CRISPR tool, is that they will be biallelic, heterozygote, or even homozygote mutations. Okay.
Yes. In terms of the chimerism, and we have to separate two parts. One is the transformants, they are chimerism. The other one is the editing is chimerism. So editing chimerism, we also talked about it, and we probably would use genetic to like filter it. And for the transformants chimerism, typically if we use morphogenic genes and we start with a low transformation efficiency, so the somatic embryo induction would automatically like filter a lot of chimerism, because like typically for the sorghum, it typically is with the morphogenic gene, you can get a shoot regenerated immediately, but without, it will stay at callus or immature embryo status for a long time. So if we do that, that's a good way using the morphogenic gene to filter out the chimerism of the transformation. Yeah.
Next, we have a general question. So what would you say would be the most promising genes for increasing plant regeneration?
So if I had to say, I will test all of them and combination of all of them. I think that so in my experience, so GRF shift works well, but we use GRF shift in a protocol that was very good already. But I mean, it has a low regeneration efficiency, but all the cell biology and tissue culture was fantastic, and GRF shift works well on that. But there is an example that we have, Andrea Galavati in maize. He combined baby boom with GRF shift, and he also observed synergy. I know that people from the University of Florida is using plethora 5, that is a baby boom like Shen, together with GRF shift with good results. So I think if it is up to me, I will test combinations and every tool that I have, and most of them are now available at Shen. And the important thing that I will mention here is, again, so we focus a lot on the morphogenes, but the agrobacterium part, and I like agrobacterium a lot, is very important too. So Greg has a lot of tools there. The ternary vectors are a great tool too. So there are many other tools that help. That's my opinion.
Mm-hmm. Okay. One of the related question is, so what would be the most effective combination of tools developed to determine the morphogen that will function effectively to transform tree species?
I think I just have to, give an answer that's very similar to Juan in that, I think you have to try multiple ones. You have to, work on the in vitro protocol. Unfortunately, transformation, you just have to go through the whole process. There's no way to kind of miniaturize it and make it more high throughput. You can try to do things like blender rather than than wound them to try and increase your throughput. But the sort of sad reality is it's still pretty low throughput. And so that's why I mentioned that it's important to just try things and look for qualitative effects first, is that, some of these are going to be really effective and they're great tools in the toolbox, but you have to do the hard work of putting this through the pipeline and figuring it out.
Mm-hmm. Thank you. We have a few more questions for Greg. So you mentioned the gene orientation is important. Is this for the 6B and 3' or for all of the morphogens in the TDA?
Yeah, so we've done some testing. Originally, the whole point was for me to try and do it in Golden Gate, and I had to make some cloning compromises with the orientation and stuff. Especially, these are big regions, so 10 kb, you're going to have like 20 BSA1 sites, right? So a lot of mutagenesis. So putting the 6B gene next to the left border where it might get truncated for sure reduces the activity. But also we noticed that when you change the orientation, the gene orientation of the auxin and cytokinin genes, it matters. Basically, I think a lot of agrobacterium are under selective pressure to make the tDNA smaller because you get increased delivery when you do that. And so that regulon, once it's inserted into plant DNA, is kind of you get variation based on position effect, but they're sort of evolved to be in that configuration. And so I'm trying not to break it when I do that, or if I am, it better be intentional. So yeah, we found some interesting things just by changing the gene order and things like that.
Okay, so we are going to finish our webinar. We're going to answer one last question directed to Greg. Have you looked into the importance of other plasgens along with the 6B?
Yeah, so 6B is part of this large gene family. They're kind of globular effector proteins that also include role B, which is really important in hairy root formation. If you look at them, they're very low sequence identity in terms of similarity. We haven't tried any other plasgenes, but we're very interested in how they function as effectors. They seem to have very disparate functions, like role B binds to topless to interfere with auxin signaling. The 6B probably binds to argonaute cerate and also may function as a chromatin remodeler. So I think these are kind of rapidly evolving protein interactor things that sort of interfere with key signaling pathways. And so I think because of that, they're probably very useful to mine for ways that they interfere with plant development. So I haven't looked at them yet, but we hope to move on to that in the coming years.
Yeah, thank you, Greg, and thank you all the panelists. Thank you, Jianqiang, Juan, Greg, and thank you for all the attendees and the PMAX team members. So once again, thank you all. We're gonna wrap up today's webinar. This webinar brought to you by PMAX, Plant Method Exchange. If you search Plant Method Exchange, you'll be able to find our website. We have a collection of protocols in there. And in the meantime, if you have protocol that you want to share with the community, feel free to send it to us our way. You will find our email on our website. And this webinar is also co-hosted by Plant Postdocs and powered by NASC. Thank you all for attending. We'll see you at our next webinar.
Thank you.